Our third line of defense is our adaptive or acquired immunity. This is going to be unique to each individual because it's going to be dependent upon what you have been exposed to. The immune system has to develop immunocompetence. That is, it has to be able to react only to foreign substances called antigens. An immunologist is someone who studies the immune response. Specific immunity shows three components. First of all, it is specific. Instead of going after just anything, it responds to a particular molecule. Second of all, we show immunotolerance. That is, the body will respond to foreign molecules, but not any molecules that are part of the self proteins. And thirdly, it has memory. Every encounter leads to the production of memory cells. After the initial reaction, you always have memory cells left. This means that the second and subsequent time you run into the same antigen, you have a much faster and more protective response. The lymphocytes are the primary cells of specific immunity. All lymphocytes are formed in the bone marrow. The B lymphocytes, or the B cells, mature in the bone marrow. They're responsible for the part of specific immunity that's known as antibody-mediated immunity. T lymphocytes, or T cells, mature in the thymus. They're responsible for that part of adaptive immunity that's known as cell-mediated immunity. Both types of immune responses stimulate and regulate innate responses. You'll remember that lymph is plasma that's left in the tissues. It's colorless, it's a watery fluid, and it's collected from the tissue by the lymphatic capillaries. This is a blind-ended system so that all of the lymph flows toward the heart. It's a one-way system. Along the way, the lymph goes through the lymph nodes. The lymph nodes filter the lymph. Since this fluid has just washed through the tissues, it's likely to have any kind of abnormal material in it. The lymph nodes then are looking for anything abnormal. The lymphoid organs are those organs that are involved in the immune system response. The bone marrow and the thymus are the primary organs since they're the site of maturation of the cells. Lymph nodes, spleen, and less organized tissues in the secondary organs like malt tissue and tonsils make up the other lymphoid organs. The secondary organs act as surveillance sites, that is, places where fluid flows through and we have cells that are looking at the fluid, constantly looking for anything foreign, and if it's found, then the appropriate cell can divide. We have proliferation of the cells. Antigens are molecules that stimulate an immune response. This depends upon their molecular shape, size, and complexity. Epitopes, or the antigenic determinants, are the part of an antigen that determines the immunological response. It usually takes fairly large molecules to stimulate the immune system. Complex molecules have more epitopes on them, or more antigenic determinants. A complex molecule may stimulate several different antigenic responses. Exogenous antigens are toxins, secretions and components of the microbial cell wall, membranes, flagella, pili, all of those foreign kinds of things. Endogenous antigens are protozoa, fungi, bacteria, and viruses that reproduce inside body cells. Their antigens are incorporated into body cell membranes so that we see them as though they're on our cells. Autoantigens are antigenic molecules derived from normal cellular processes. Let's look first at the T lymphocytes. Their maturation involves developing T cell receptors. Now, the body can make thousands of different receptors. Each one is for a different antigen. We have the ability to respond to hundreds of thousands of different antigens. T cells are not going to bind directly to the epitopes or the antigenic determinants. This means that those antigenic determinants have to be associated with the major histocompatibility complex proteins that are found on every cell. Your major histocompatibility complex proteins are unique to you. They're what we refer to as your self-proteins. Antigen-presenting cells, or APCs, have major histocompatibility complexes that can act to present an antigen to a T cell. Macrophages, dendritic cells, and B lymphocytes have these kinds of MHCs. So the T cell receptor will be a protein on the surface of the T cell that will have an antigen binding site. Each T cell will have a different antigen binding site. All cells except red blood cells have major histocompatibility complex type 1 proteins. This is part of our self-recognition. Antigen-presenting cells also have an additional set of major histocompatibility complex proteins. These are called MHC2 proteins. These are the ones that are involved in regulating the immune response. Antigens have to be processed by the antigen-presenting cell and have to be placed in that MHC complex in order for the T cell to see it. Antigen-presenting cells will then dock with the appropriate T cell receptor and stimulate that T cell to activate. So we have class 1s on every cell. They have these grooves, these antigen binding sites. Class 2 are found only on antigen-presenting cells. They look a little different, but they still have an antigen binding site. It is only this receptor, however, that the T cell can bind to. The T cell can't bind to this one. There are different types of T lymphocytes, and these are determined by the glycoproteins on the cell membrane and by their characteristic function. The cytotoxic T cells have the CD8 receptor on them. Cytotoxic T cells can directly kill other cells containing the specific antigen to which they react. Now these are different from natural killer cells. Natural killer cells just attach to and kill anything that's not self. Cytotoxic T cells are going to have a specific target that they're looking for. The T helper cells are the CD4 cells. They're involved in regulating the function of the cytotoxic T cells and the regulatory T cells. There are type 1 helper Ts. These are the ones that assist the cytotoxic T cells and help regulate innate immunity. The type 2 helper T cells function with the B cells, fully activating them to produce antibody. T cells secrete a group of chemicals called cytokines, and these cytokines vary, but they help regulate the immune system. The regulatory T cells have CD4 receptors on them and CD25 receptors on them. These are the cells that help repress the adaptive immune system. They're also involved in preventing autoimmunity. T cells have to go through an education process. As we're forming T cells, we make those that will react with self as well as those that will react with non-self proteins. We need to make sure we eliminate those that react with self proteins and only keep the ones that react with non-self proteins. They have to go through 2 tests. Because the T cells have to recognize MHC proteins, we want to make sure that all of the T cells recognize self-MHCs. So we have them dock up, and if they don't dock with a self-MHC, we get rid of them. We next have to check for those that recognize self-antigens in that MHC. When there are self-antigens in the MHC, if the T cell recognizes it, we want to destroy those. That is what provides us with what's known as self-tolerance. We learn to tolerate all of our proteins. These cells are said to be naive. They have not encountered their actual antigen. In order to be fully mature, they have to encounter their specific antigen. We have many T cells that will never encounter their antigen. They will always be naive T cells. B lymphocytes have to go through a similar process. The B cell receptors code up the B lymphocytes just like they do on the T cells. The B cell receptors look a little different. They're immunoglobulins that are embedded in the cell membrane. Immunoglobulins are 4 protein chains. 2 of them are long and 2 of them are short. The long ones are called heavy chains and the short ones are called light chains. They have a constant end that's embedded in the membrane and the variable end. The variable end is the antigen binding site. When an antigenic epitope or an antigenic determinant stimulates a B cell, we will get an activated plasma cell. It's the plasma cells that produce the antibodies. So the receptors on the B cell look a little different. Here are the heavy chains and the light chains. These are the constant regions. They're the same for every one of these. It is these variable regions out here that will be different. These will be the ones that will recognize a specific epitope. Again, we will make thousands and thousands and thousands of B cells to respond to all kinds of proteins. Only a few of them will ever actually encounter their protein and be activated. Antibodies are free immunoglobulins that circulate in the plasma. Basically, they have the same Y shape as the transmembrane receptor. The arms contain the antigen binding site. The stem is constant in each antibody class, and we have 5 antibody classes: IgA, IgM, IgE, IgD, and IgG. IgM is the first responder. It is the first antibody that's made whenever a B cell encounters its antigen. It's very effective at binding complement. It is the largest of the antibodies. It's 5 of those Y-shaped units held together in a circle. IgG is your most common immunoglobulin. It only has one Y-shaped unit. IgG is small enough to cross the placenta. It plays a major role in all aspects of the immune response. Whenever you have a second or subsequent encounter with an antigen, the memory cells are going to be stimulated to produce IgG-type antibodies. IgA have 2 units. Now, IgA is called the secretory immunoglobulin. We find this on mucous membranes. We think it may act to bind to materials that try to come through the mucous membranes. IgA can also be passed to the infant from the mother in breast milk. IgE is a single Y unit. We find this one coating up mast cells. IgE plays a role in allergies. And the IgD is also a single Y unit. This one is bound to the membrane of B cells. We know that plenty of mammals don't have IgD and still have a very functional immune system, so we don't really know the role of IgD. Antibodies function to either inactivate or neutralize their target antigen. They do this by activating complement and initiating inflammation, neutralizing toxins or blocking receptors that neutralize viruses. They can enhance phagocytosis by coating up a foreign cell, allowing a phagocyte a better opportunity to eat it. Some of them can kill by oxidation. That is, they catalyze the production of hydrogen peroxide or ozone or other oxidants and kill bacteria directly. They may cause the antigen to agglutinate. Now the phagocytic cell has a much bigger target to ingest, so we take more organisms out with each phagocytic encounter. Antibody-dependent cellular cytotoxicity happens when antibodies coat target cells and then bind to natural killer cells. The natural killer cells will then release perforin and granzymes and kill the cell that the antibody is coated on. So as we look at some of these types of things, neutralization, we block it up so that it can't be effective. We enhance phagocytosis by making it easier for the phagocytic cell to find it. We may actually kill it outright by causing oxidizing agents to kill the cell. Here you see several antigens bound together. Think of a phagocytic cell encountering this. Instead of eating each individual cell, it can take 6 at one bite. And this is the coating up of a cell so that a natural killer cell can bind to it and kill the effective cell. B cells also have to be educated. They go through a similar selection process as the T cells, but we don't understand this process nearly as well. But we do have to eliminate those that react with self so that we have self-tolerance, and we have to keep those that react with non-self. Again, these educated cells are still naive. They only mature when they're activated by their specific antigen. Many of these cells will never be activated. There are several chemical mediators of the immune response. These are generally known as cytokines. These are regulatory proteins that can act as intracellular messengers. They're released by certain body cells. Mostly they're concerned with signaling among the immune leukocytes. Interleukins are one kind of cytokine. They signal among leukocytes. The gamma interferon that we talked about earlier is involved in activating phagocytes. Growth factors will stimulate leukocyte production. These have to be released in order for us to get leukocytosis at the beginning of inflammation. Tumor necrosis factor can be secreted by macrophages and T cells. This will target and kill tumor cells. It also helps regulate immune responses and inflammation. And chemokines are chemotactic factors. They help call the cells to the appropriate area. Let's look at the cell-mediated immune response. In this response, the antigen has to be processed by an antigen-presenting cell and presented to T cells. Now it will go around cell by cell finding the correct T cell, because there's only one T cell that's going to respond to this particular antigen. Think about it as Prince Charming looking for Cinderella with the glass slipper, except there's about 20, 30, 40,000 different Cinderellas he has to try this glass slipper on. The CD8 or cytotoxic T cell can respond here. There has to be a nearby CD4 cell to assist this process. The CD4 cell is going to recognize that same antigen that the CD8 cell recognized. The CD4 cell will bind to the antigen presenting cell at the other MHC site, so that we'll have an antigen presenting cell bound to a CD8 and a CD4 cell. The CD4 cell will release cytokines. These cytokines are necessary to fully stimulate the CD8 cell and make it responsive. The CD8 cell is now mature. It has met its Prince Charming. The glass slipper is on its foot. It now knows exactly what protein it has to go out and find. This CD8 cell now clones. It makes multiple copies of itself. After it clones, the daughter cytotoxic cells can start to reproduce without being attached to the antigen presenting cell. We call this self-stimulation. The cytotoxic T cells now get into the circulation, and they circulate looking for the cells that have that protein that they are targeted to destroy. They secrete perforins and granzymes, which destroy the cell once they attach to it. Cytotoxic T cells have to directly bind to the cell that they have to kill. Also, when we're cloning all of those cytotoxic T cells, we make a few memory cells. These cells are going to live in the lymphoid tissues, and they'll be available should we encounter that particular antigen in the future. Here we see a cell, an antigen-presenting cell, that has found an antigen, and it's placed it in its MHC1 because the cytotoxic T cells can bind to that. But that's not going to fully activate this cell. We have to go ahead and find the appropriate CD4 cell that will bind to the MH2. We're going to have interleukins released. These interleukins are necessary to co-stimulate the CD8 cell. Without this, the CD8 cell will not become effective. So our T helper cell starts making these interleukins. We fully activate the CD8 cell, and it clones. We make a memory cell or two. It's going to hang around in the lymphoid tissues for later if we need it. But we're going to clone a whole bunch of these CD8 cells that are going and looking for this particular epitope, this particular antigen. Whenever it finds it, it will attach to the cell that has that, and it will kill the cell. T cells require several signals in order to respond to the antigen presenting cell, and we have to get all of those signals in the appropriate sequence. If they come out of sequence or if they don't come, then there is not a full response. This helps prevent autoimmunity, accidentally triggering a response when we shouldn't. Regulatory T cells are involved in moderating cytotoxic T cells. We don't understand the role of regulatory T cells well, but typically T cells begin to die off in about 8 days, and they're totally gone in about 30 days. The other type of acquired immunity is the antibody immune responses. We have T-independent antibody immunity. There are a few antigens that have numerous repeating subunits. They can directly trigger B cell response without using a T cell mediator. This will initiate the production of plasma cells and antibody production. The response is very fast, but it's also very weak. None of the cytokines are produced that are necessary to fully develop the B cell. Seldom are memory cells developed as a result of this response. Here you see a large molecule with lots of repeating subunits. They can bind directly to the B cell that recognizes this particular antigenic determinant. The B cell will clone and we will make a few antibodies. But notice that we only get plasma cells. We don't get memory cells. So this is not a very effective way to stimulate antibody-mediated immunity. Most antibody immune responses are T-dependent antibody immunity. T-dependent antigens lack numerous or repetitive or identical epitopes. The antigen binds to the B cell or some other antigen-presenting cell, and the antigen is taken in by receptor-mediated endocytosis. The antigen is processed and placed in one of those MHC2 binding sites. Then it can be presented to a T helper cell. Cytokines then signal the T helper cell to convert to a type II helper cell. This is the kind that interacts with B cells. The T helper binds to the appropriate B cell, produces cytokines that stimulate the cloning of the B cell to plasma cells that produce antibodies and memory cells. So here we have an antigen-presenting cell finding the right CD4 cell. This CD4 cell clones a little bit, starts making its interleukins, and differentiates into the helper T, and differentiates into the type II helper cell. The type II helper cell goes out and finds the right B cell. This stimulates the B cell to clone into plasma cells. We're making a lot of IgM initially and a little bit of IgG. And we make some memory cells that now have that particular receptor for that particular antigen. Memory B cells do not secrete antibody, but they can survive long periods of time, 20 years or longer, and they can be activated again if needed. The primary response is a little bit slow. First of all, there's a lag time of about 3 to 5 days as we're finding the right T helper cell, converting it to the type II helper cell, finding the right B lymphocyte, doing all of that cloning. That takes a little bit of time. We produce primarily IgM, and this peaks at about day 7. We do produce larger amounts of IgG, but that peaks at about 2 weeks. This response usually does not occur fast enough to prevent the person from acquiring the disease. However, the secondary response, the subsequent response, almost as soon as you encounter that antigen again, there is an immediate antibody production. The memory cells are stimulated, we get large quantities of IgG, we get a little bit of IgM, but we get a much more rapid response and we get a greater response. So if we look at the primary response, there's the lag period when nothing much is going on in terms of antibody production, but we're doing all of that cell activation. We get that IgM that is usually what gets us over the disease, and we get IgG at about 2 weeks. The next time we see that same antigen, almost immediately, we get IgM, but we get a huge amount of IgG. Not only do we get more, but it persists longer. This is now protective. There are different types of acquired immunity. In active acquired immunity, you make the antibodies and you're going to have memory cells. So this is more or less a permanent kind of immunity. Naturally acquired active immunity means that you are actually exposed to the antigen. This typically means you have had the disease. Artificially acquired active immunity is the kind of immunity you get from an immunization. You encountered the antigen, you made your own antibodies to it, but you did not come across this antigen naturally. You were deliberately exposed to it. In passive immunity, you get antibodies from some external source. Because these are proteins, they degrade over time, and you didn't make them yourself, so you have no memory cells. So this is a temporary kind of immunity. Naturally acquired passive immunity is the immunity a mother passes to its child. Remember, those IgG antibodies can cross the placenta. Also, if the child breastfeeds, IgA antibodies can be transmitted from mother to child in breast milk. Artificially acquired passive immunity is when you are given some sort of anti-sera or antitoxin. It gives you immediate protection, but it does go away, and should you encounter that particular antigen again, you would have to have anti-sera or antitoxin again. So as we look at our summary of host defenses, innate defenses or inborn defenses are first line or barriers, physical, chemical, genetic. Our second line of defense are things like the inflammatory response, interferons, phagocytosis. And then our third line of defense is the acquired immunity. This is based on what we come in contact with, and we can have natural or artificial immunity. Immunization is a means by which we can get active immunity without having to wait around to have the disease. In the 12th century, the Chinese noticed that survivors of smallpox never got the disease again. They began the process of variolation. That is, they took scabs from someone who had smallpox and they ground them up and they inhaled them. This had a little bit of danger to it. Most of the time nobody got too sick, but sometimes people would get sick and die. In 1796, Jenner noticed that milkmaids had beautiful complexions because they never got smallpox. He discovered that they were exposed to a form of smallpox found in cows, cowpox. And because the two viruses were so similar, being exposed to one gave you immunity to the other. So he developed a vaccine based on cowpox. This was how we overcame smallpox initially. As a matter of fact, the word vaccine comes from his word. Vacca is Latin for cow, so vaccine was something that we got from a cow. Pasteur further refined vaccine production and came up with some other ways to come up with organisms that could be used as vaccines. Active immunization uses several different types of vaccines. We always check the effectiveness by checking the antibody titer in the blood. That is, we look at the level of antibody that's been produced in the blood. If the titer is low, we need to re-stimulate the memory cells. This is the purpose of a booster shot. There are several different types of vaccines. An attenuated vaccine or a modified live vaccine is when you're injected with an organism that's been weakened to reduce its virulence. The organism can no longer cause disease. Now, sometimes you do get a mild infection, and vaccinated individuals may be carriers for a short period of time. You do not want to use this kind of vaccine in someone who's pregnant or immunosuppressed. That mild infection could be rather dangerous for either of these individuals. Inactivated vaccines use killed organisms. Whole agent vaccines give the entire organism, but it's dead. And subunit vaccines use antigenic fragments, just pieces of the dead organism. These are sometimes administered in high doses. Sometimes there have to be repeated doses with an adjuvant or some sort of helper that helps increase the antigenicity. One of the problems with this is the adjuvant may act as an allergen or may cause local inflammation. Toxoid vaccines are chemically or thermally modified toxins. Toxoids have very few antigenic determinants, so when you're giving a toxoid vaccine, usually you have to have repeated doses. So you have several doses in childhood and then boosters every 10 years or so. In combination vaccines, they combine antigens from several different toxoids and from inactivated pathogens. The MMR, the DPT, are examples of combination vaccines. They now have Pentacell, which is diphtheria, pertussis, tetanus, polio, and Haemophilus. Then we have newer vaccines that use recombinant gene technology. These are safer. You can make large quantities of these vaccines, and we can also make vaccines to things that we've been unable to vaccinate before, like fungi. Here is the CDC recommendation for immunization schedules in individuals for all of these different organisms. You can see some of them are one dose, most of them are repeated doses, so that we continue to stimulate memory cells every once in a while and make sure that they don't lose their memory. Passive immunotherapy is administering antibodies to a patient when we need to protect the patient quickly, when the immune system cannot respond fast enough to the agent to provide protection. The antitoxin for botulism is such a case. The botulism toxin kills pretty quickly, so you don't really have time to get over it, develop antibodies, and then wait for a second case to be protected. The first case will kill you. Antivenom is also given for snake bites for much the same reason. We can use antigen-antibody reactions in diagnostic testing. We can determine if someone has been exposed to a disease by testing for antibodies in the blood. Basically, any of these tests require us to mix the patient's blood with known antigens to see if we get any kind of reaction. We can use this to determine if an immunization has been effective. We look for the titer of the antibody or the level of the antibody to make sure it's high enough to be protective. We can use this to diagnose current or past diseases by seeing if antibodies are in the patient's blood. There are a variety of testing techniques. We check for all of the things that antibodies typically do when exposed to an antigen. We look for precipitation or agglutination or neutralization, some way to produce antigen-antibody complexes or to fix complement. We can pretty much coat any kind of particle with the antigen and then put that with the patient's serum and see if there's any kind of reaction, an agglutination reaction, a precipitation reaction. We can also tag antibodies with fluorescent dyes. We can use this for direct staining. We can also use this for enzyme-linked immunosorbent assays or ELISAs. Point of care testing is this kind of testing for quick diagnosis, but no quantitative analysis can be done. Pregnancy testing and rapid strep testing are examples of point of care testing. You simply get a positive or a negative result.